Hey everyone, and welcome to my channel. Today we're going to have a look at the MAP kinase pathway. This is one of the most important pathways in cell development and cell growth. And today we're going to have a look at the proteins that are involved in this pathway. We're going to have a look at ligands such as growth factors, and then we're going to have a look at some small molecules that have been developed to target this pathway for therapeutic uses. But let's start by the name. Why is it called MAP kinase pathway? MAPK. So, MAPK. MAPK stands for Mitogen-Activated Protein Kinase. Okay, so what is a mitogen? A mitogen is a substance, such as, for example, here, a ligand, that promotes mitosis. So mitogen is a growth factor because it causes cell to divide and grow. Activated protein kinase. So this is MAPK pathway. So it's a pathway that involves some protein kinases that, as we will see, are proteins involved in the multiplication of the signal inside the cell. Because as with many pathways, we have protein kinases that act like, I don't know if you know, photomultipliers. So a photomultiplier gets in a photon, and then the photon hits the photomultiplier, and for one photon, for example, we have doubling of the signal, and then four signals, and then signal multiplies. And so this is the role of the kinase cascade here, RAF MEK ERK, because this causes for one molecule, for example here ligand, we have a big signal that can be produced inside the cell. But let's start by the ligand. So as we said, the ligand is a mitogen, and in this case we can have, for example, EGF, epidermal growth factor, but we can also have different other growth factors such as FGF and PDGF, but the proteins can vary a little bit. But this is the canonical MAP kinase pathway with GRB2 SOS. Some other pathways may differ in some proteins, but the concept is more or less the same. They all have a receptor tyrosine kinase receptor. As we said in the previous videos about receptor tyrosine kinases, these are receptors that dimerize once the ligand is bound to the receptor. So they dimerize and then they autophosphorylate. So this means that the receptor tyrosine kinase, once dimerized, puts a phosphate in the other receptor tyrosine kinase, and this in a tyrosine, and then we have the recruitment of SH2 or SH3, I don't remember exactly, proteins such as GRB2 and SOS. These are adapter proteins. So here we have a zoom inside the RTK, and so we have the phosphorylated tyrosine. Here, for example, here we have the phosphorylated tyrosine attached to the receptor, and we have GRB2 that recognizes this phosphorylated tyrosine and binds the receptor. GRB2 connects to SOS, and SOS activates RAS. So RAS is a GTPase, so this means that it's a protein that's coupled with GTP, and it's embedded inside the membrane. As we can see here, we have a little tail that's embedded inside the membrane. So RAS GDP is inactive and RAS GTP is active. As with many GTPases, also in the adenylate cyclase pathway, it's the same. So SOS promotes nucleotide exchange, because if we have to pass from GDP to GTP, we have to exchange a nucleotide. If we have to inactivate RAS and to pass from GTP to GDP, we can hydrolyze a phosphate like with ADP. So here we don't have a nucleotide exchange, and here we have a nucleotide exchange. Here we have nucleotide exchange. Now, RAS is a very important protein, and it's involved in many cancers, including, I think, melanoma, for example, because here we have EGF, epidermal growth factor, and melanoma is cancer of the melanocyte in the skin. So epidermal is the part of the body that has the cancer, epidermal tissue. Now, so RAS, really important to remember RAS protein. So when there are also many inhibitors that have been developed for RAS. Some work, some don't work, but it's an active area of research of RAS inhibitors, small molecules to inhibit RAS or antibodies. Now we have now we can have the cascade that we talked before. So we have RAF, MEK, ERK, and RAF phosphorylates MEK and MEK phosphorylates ERK. So these are also called MAP. RAF is also called MAPKKK. So MAP kinase. And MAP kinase is a kinase that phosphorylates, so here in red, MAP kinase phosphorylates MAPKK, and MAPKK phosphorylates MAPK. And then MAPK is, or ERK, is responsible for different gene regulations inside the cell. So we have activation of some transcription factors, modification of transcription, and including the transcription of G1 to S cyclin, so cyclin D, CDK4 or 6. This complex, if you remember, is responsible for the phosphorylation of PRB. If you draw, this is PRB, and PRB inhibits E2F. This is E2F. So once cyclin 4,6-CDK complex are present inside the cell, they phosphorylate PRB, and PRB releases E2F. E2F now can go and promote synthesis of DNA, and so we can enter the S phase. So this is a little bit of one of the mechanisms by which this pathway can promote cell division and multiplication and growth and mitogens, etc., etc. Now, I hope this part is clear. This pathway is quite simple. So we have RTK, phosphorylation, GRB2, SOS, RAS, GTP to GTP, RAF, MEK, ERK, and then gene transcription regulation, etc., etc. Now, what I told you that this pathway is involved in some diseases, including cancer, because it's a pathway that involves growth factors, and we know that cancer tends to grow a lot. Now, there are some small molecules that have been developed to target this pathway, and one of these small molecules I wanted to show you is let me go down, is this one. So dabrafenib. Dabrafenib is an inhibitor that targets RAF. So this molecule right here targets RAS. And you can know molecules that target this pathway, I think RAS and RAF, because they end by IB or NIB. And so this is a small molecule that I think is in commerce for treating of melanoma or something like that. So I hope this video was informative and clear. If you like this video, please give me a thumbs up and maybe subscribe to my channel to have more videos about biology. I hope you have a great day, and I hope to see you soon. Bye bye.